Today, we will be talking about peeling away uncertainty, a probabilistic approach to DNA mixture deconvolution. The concept is that by successively conditioning on genotypes, we can get more identification information from DNA evidence. This work is based on a master's thesis conducted by Chaudhry under the supervision of Dr. Mark Wilson, using data from John Donahue's crime lab. DNA mixtures arise when multiple people contribute their DNA genotypes to the same evidence item. Here, in this example, we see 5 different people, all of whom are related, leaving their DNA together in one mixed sample. Their genotypes are combined, and then when the laboratory does PCR amplification and size separates the data with fluorescent detection, we obtain phenotypic data of the short tandem repeats, or STRs, at each tested location. As we see in this STR electropherogram, there are fewer peaks than you might expect from 5 unrelated people because of sharing of alleles. The genotype unmixing problem is the inverse problem. How do you go from the phenotypic data that's produced from the individuals back to the genotypes that produced the phenotypic data? Because of genotype uncertainty, the genotypes of each of the 5 individuals can only be known up to probability. Uncertainty arises from the PCR experiment itself, which is a random process and is different on every amplification. And uncertainty in this case also arises because of the allele sharing between the genotypes of the related family members. Identification information is measured by comparing an evidence genotype with a reference genotype relative to a random population. That comparison is quantified in one number called a likelihood ratio, abbreviated LR. The likelihood ratio measures the change in genotype probability made by the DNA evidence data. Prior genotype probability reflects a random population. The posterior genotype probability is how the data has concentrated probability on more likely genotype values. The ratio of posterior to prior genotype probability gives the likelihood ratio. The logarithm, or powers of 10, of the likelihood ratio is a standard information measure in science.
Genotype uncertainty is seen as a diffuse probability distribution that spans over several allele pair possibilities. It generally derives from contributor overlap, where DNA contributors are incompletely separated due to low DNA amounts or biological relatedness. The more uncertainty that is present in genotype inference would result in less match information. So decreasing this uncertainty can lead to more informative match statistics. But how can we go about doing that? Well, we can do this by using a process called genotype conditioning or genotype peeling. So in genotype inference, conditioning assumes a statistically established contributor genotype. The conditioning process is performed iteratively and in subsequent analyses of the same mixture data. So within each analysis, a contributor genotype is assumed using known reference samples that remain constant throughout a computer run. So this contributor's genotype becomes locked in, and the inference is then focused on the remaining unknown contributor genotypes. Because probability distributions are then refocused on the remaining unknown contributors, conditioning can improve genotype separation by reducing the uncertainty in these other unknown genotypes. With this reduced uncertainty, match information improves, whether inclusionary or exclusionary. So here's how the peeling process works. Initial mixture separation is performed where all contributor genotypes are inferred with no assumptions. This establishes the most informative DNA contributor. In a subsequent computer run, this statistically established contributor is conditioned on, and mixture separation is then repeated under this assumption. Multiple rounds of processing peels away the unknown contributor genotypes until only one remains. In our study, mixtures were also separated using the zero unknowns feature in TrueAllele, where contributor genotypes are assumed, and the computer can then determine highly accurate mixture weights since genotypes are certain. To show the effectiveness of conditioning, mixture samples comprised of 2 through 5 contributors were examined. Mixture composition followed a randomized design in terms of genotype and DNA amounts. Samples were amplified using the Global Filer PCR kit and then size separated and detected on the 3500 genetic analyzer. From there, sample data were then uploaded to TrueAllele's cloud server, and computer runs were set up using an expected number of contributors and were processed for 25K Markov chain Monte Carlo cycles. Runs were also created in duplicate to view reproducibility, and additional cycles and replicates were conducted as needed to achieve concordance. In genotype inference, mixtures were conditioned based on the highest match statistic, and unknown contributor genotypes were peeled away until only one remained. Inferred genotypes were compared to 70 references to calculate LR match statistics. So taking a look at some of the data from this study, these graphs show how subject 34 matches in this 5-contributor mixture by focusing in on one locus, D1S1656. The graph on the left shows the genotype probability distribution, and the right shows the respective locus log LR values. Under genotype view, the blue bars correspond to the inferred genotype probability before conditioning starting at Q3, followed by 4 successive peeling rounds, and the locus log LR values are displayed in the same order in match view. Before peeling, genotype probability distribution was more spread out over several allele pair possibilities, and this was because of the high levels of uncertainty present, which resulted in a negative log LR value. As conditioning progressed, uncertainty was peeled away, and the probability distribution became more concentrated at allele pair 15-16.3. With that reduced uncertainty, log LR values show incremental improvements as conditioning progressed, just at this one locus. Now, this is just looking at one contributor in one mixture at this one locus. And we know that the total match information per contributor is determined by adding together these independent locus log LR values per peeling round. So let's take a look at that. So this is the same 5-contributor mixture that we've seen previously. This table shows the total match information per contributor after successive peeling rounds. The most informative contributors with log LR values highlighted in green were conditioned on in the subsequent run. In general, the log LR information increased linearly with contributor DNA amounts until reaching a maximal information plateau for major contributors. So taking a closer look, this mixture contained two 11% components, contributors 30 and 34. Initially, the likelihood ratios for these overlapping contributors were in the thousands. Peeling these contributors away from the major contributors increased their LR values into the billions, and peeling the two 11% contributors away from each other increased the LRs to almost a quintillion. The results of this study conclude that genotype conditioning improved separation for overlapping contributors and overall doubled their match information.
This case example of the Queen versus Robert Z is from Australia. On the night of the 18th of July, 2009, in a Sydney suburb, 5 Lin family members were bludgeoned to death in their North Epping home. There was blood splattered throughout the house, often a mixture of the different victims of the crime. Suspicion fell on the wife's brother-in-law, Robert Z. Z had cleaned his garage floor that morning after the murder. The police examined many stains on his garage floor. This is blood stain 91. In the left figure, we see an arrow pointing to a small, short streak. That is the stain that contains DNA from multiple individuals. On the right is an electropherogram at the FGA locus showing peaks from what looks like to be around 3 individuals. However, given the relationship between the victims, there could be allele sharing, and this might be 4 or 5 different individuals. Here we see the Lin family, the 5 victims. The father, Min, age 45, and his wife, Yun Lee, age 43, and to the right, her sister, Yun Bin, age 39. On the bottom row, we see the 2 children, sons Henry, age 12, and Terry, age 9. The forensic question is there DNA from the Lin family members in the garage floor mixture at Z's house? If so, that could indicate that Z had brought the blood back on his shoes from the crime scene into his garage floor and his house. Without peeling, all 5 relatives look to be associated with the garage floor stain. Is this due to kinship, allele sharing, or is their DNA truly present? TrueAllele gave a peeling answer. Looking at this match table, calculated at a 3% coancestry coefficient value, we see a comparison between the garage floor DNA separated genotypes and the 5 reference samples. In the first row, without assuming any conditioned genotypes, we see in yellow that son Terry has a log LR value of about 13 or 10 trillion. He is associated with the stain. In the second row, conditioning on Terry's genotype, we see highlighted in yellow a log LR value of 6.9, which is around 10 million, between the garage floor stain and son Henry. The third row, conditioning on Terry and Henry's genotype, produces a match statistic of 9. A log LR of 9 corresponds to a stat of about 1 billion to the father, Min. So he's also present statistically in the stain. Continuing the peeling in this way, we see that the mother, Yun Lee, is not statistically associated with a garage stain, whereas her sister, Yun Bin, is. So with peeling or repeated genotype conditioning, TrueAllele has established that 4 of the family members are clearly present in the garage floor bloodstain. A second forensic question is whether sister Brenda Lin's DNA is present in the garage floor mixture. It shouldn't be, since the 15-year-old girl was over 1,000 miles away in New Caledonia on a school trip. However, without peeling, Brenda Lin might be present due to the DNA match statistics. Is this because of sibling allele sharing, or is her DNA truly there? Conditioning on the genotypes of her 5 family members reveals the truth. In the bottom right of this log LR table, we see a value of -6, or an exclusionary value less than 1 in a million. With peeling, Brenda's DNA clearly is not in the garage floor stain. In conclusion, mixture data contain DNA identification information. Comparison between DNA evidence items can be made through their genotypes. The likelihood ratio measures match information between the items. Genotype conditioning can increase that match information. Successive peeling rounds of genotype conditioning reveal DNA matches that otherwise would not have been known. Our laboratory studies demonstrate the efficacy of this approach, and a criminal case application shows its utility. Better use of DNA data can give more identification information. Thank you.